All right, guys, you want me to depress you even more and get you all paranoid and begin to question everything in your life? Well, I hope that I can do that. I mean, that's one of my goals, right? I mean, this is the kind of stuff that I want you to do all the time. Second guess everything. So, our next case is going to be good old-fashioned Tylenol. Oh, that's right, good old-fashioned Tylenol. What was going on with Tylenol? Oh, no. You mean I go to the store, I buy an over-the-counter drug product, I take it home, pop the cap, the foil cap, and I take out one of those tablets and I swallow it? And I should second guess what was going on with Tylenol? Oh, well, see, I was very clever in the way that I explained that. I told you a foil cap, right? Oh. That wasn't always a thing. Go figure. What would make common sense just wasn't really getting done up until this point. So in 1982, Chicago, of all places, Chicago, someone went in, opened up some Tylenol bottles, and they dumped out some of the Tylenol tablets that were inside of those bottles, and they replaced them with cyanide pills. Cyanide pills, folks. Cyanide pills that will kill you, make you foam at the mouth. Good old Adolf Hitler. I don't know if good old is actually a proper term to describe him, though. And was he really poisoned with cyanide, or was it something else? Ooh, conspiracy theories. So, anyway, cyanide tablets were replaced with Tylenol. People would purchase the Tylenol bottles. They went home. They swallowed the Tylenol, thinking it was Tylenol, but it was not. They were cyanide pills. And 7 people ended up dying because they did not tell the difference between a Tylenol and what was not Tylenol. Because they assumed, they put trust in the process. And here, that trust in that process, no, sorry. Come again. Even creepier, the murderer, the person that replaced some of those Tylenol tablets with cyanide tablets, they were never found. So, what happened? Well, the company that manufactured Tylenol was like, listen, what we can do based on suggestions from the FDA is that we can actually put in place guidelines on our bottles to ensure that they are tamper-proof. So, foil cap. And if the foil cap has been punctured, do not take. I mean, have you read those statements on those bottles and those types of products? If this package has been opened, do not take it. I mean, that's on food products, it's on drug products, it's on many things, right? Well, the same thing's happening here. I mean, listen, I'm going to tell you the type of person that I am. If I go to a hotel and it is a swanky hotel where everything is so nice and prim and proper, and I go into the shower and there's all of these little shampoo bottles and conditioner bottles that are set out for me because maybe I didn't bring them on my real fancy trip. And I go to those shampoo bottles and I open up the bottle and there's not a foil cap, I don't use it. Because you know where my mind goes? My mind goes is, what if someone put Nair in that shampoo or conditioner? And then here I am lathering all over my head, and then I let it sit, and then all of a sudden it starts burning my hair and my hair falls out. Oh my God. Yeah, don't trust anything. So, if these seals are tampered with, or if they're opened, don't use the product. And this is one of the reasons. So, this tamper-proof packaging was actually so effective, it became an industry standard that began to get enforced by the FDA. So, here we go. Laboratory testing. We should be testing these batches, and here the batches were actually getting maybe tested and everything was going okay. But then by the time it got out to the consumer market shelf, well, tampering began to happen there. After it left manufacturing, after it left the laboratory area, it began to get tampered with on the consumer shelf. Pills were switched out. Whose fault? Whose responsibility is that? No one's really. But there's got to be a way that we can prevent that from happening in the future. So the manufacturing side got dinged. And the manufacturing side had to figure out a way in order to prevent this problem from happening in the future. Next up, I want to talk about a company called IBT. Industrial Biotest Laboratories. What a mess this place was. I'm not going to really go into too much detail here because I want you to read the article. Industrial Biotest, or IBT Labs. Guys, everything that we've talked about with good laboratory practices gets thrown out the window here. Everything. You talk about a nutcase. IBT was one of those places. There were issues with the laboratory areas because we're really focused on GLP. There were issues with the data that was getting generated by the laboratory personnel because we're really focused on GLP. And there were problems with sponsors that was using the data in some cases knowingly know what they were doing. And we get issues there as well. So in the online platform, you're going to see an article, and this article is going to be one of your assignments. And that article is going to focus on IBT Labs and what these people were doing. And again, I'm not going to maybe spoil too many of the beans for you, but I do want to give you a little bit of cover here just to get you interested in, oh my God, what is this about? Let me go to this article and let me read it. I hope that's what you do. Up at the top, faking it. The case against Industrial Biotest Laboratories. Editor's note, this article is from the blah, blah, blah. But it says, it is not an easy article to read, and may indeed be one of the most disturbing things that you have ever read. It chronicles the scandal that destroyed the credibility of the safety testing lab industry in the late 1970s and early 1980s, particularly Industrial Biotech or Biotest Labs of Northbrook, Illinois. The article reveals the fraudulent practices of IBT and other labs, the horrendous treatment of animals, and the total disregard of human health and the integrity of the regulatory processes. Many of the products, the safety of which was declared falsely, are still on the market, and this includes the notorious naproxen. But you would know that as Aleve. That's right. During the time of IBT, IBT was generating fraudulent data. The animal treatment was horrendous. The human health they cared nothing about, really. They were generating all of these reports and data, sending it to get approval on certain products that they were testing. And one of those products was called naproxen, slightly under a different name at the time. And naproxen's data was ching approved by the FDA based on what they saw, and naproxen was released to the consumer shelves, and people began to take Aleve. In the beginning, though, they were prescribed Aleve because this was a prescription medication by your doctor. So your doctor would prescribe naproxen or Aleve. You would go home, you would take this prescribed medication, and off to go. Yay! My pain, my dull and achy pain in my legs or my joints or my back, all of it goes away. Great. Worked out in everybody's favor. Did they get lucky? Maybe. Because this was based on fraudulent data by the IBT. And it was approved and it was sold. And many, many years later, they found out this. Uh-oh. Woohoo! But at that time, been released out to market. It's been out there for a while. I think we're going to be okay. Well, additional testing was done sooner or later. But, folks, this could have been another disaster. So what begins to happen here, especially with the IBT and all of these late cases, is the structure of the laboratory environment. So this was before good laboratory practices were put in place. And this was kind of the model that was going on at the industrial biotest laboratories. So what was happening here, we get a test facility, and the test facility was IBT. So I want you to think of this as like an independent laboratory company. You go to work for them, you're doing a job, and you're getting paid. This is where you get your paycheck. Well, a sponsor, these are maybe research labs that have developed this new product, and this new product needs some testing, needs some laboratory work before it can get approved. So the sponsor will hire these testing laboratories, and they'll say, hey, listen, why don't you go to this? Why don't you test these things? This is what we're looking for. Just make sure it looks good and send us the data. We don't have the time for this, but we're going to hire your company in order to do this work for us. And a company at the test facility will be like, okay, and then they'll take the paycheck and they do the testing. So they generate all this data, and the data is given to the sponsor. And the sponsor reviews the data, and the sponsor says, whoa, yeah, this looks great. A+ for us. All of this looks really, really good. Thank you, test facility. And they submit that data to a regulatory agency. And that regulatory agency can be anybody, FDA, EPA, any other regulatory agencies that are out there, depending on the product that's getting made. So in the case of naproxen, naproxen was given to IBT Labs. IBT Labs was generating data, doing some tests. They gave the data and the tests back to the sponsor. The sponsor said, great, looks wonderful. Great job, Industrial Biotest. You did wonderfully. And then they submit that data to the FDA in order to get approval. So the FDA then looks at that data that's been provided to them, because they're not part of the pipeline. They were not there. They were not in those labs. They were not at the sponsors. The FDA just reviews data, and they look at the data that's been done by a lab, who they do not know, in a location which they do not know, in an area where the testing's done that they've never visited. And they look at this and say, whoa, yeah, looks great. Check mark. Absolutely. You get approval. So naproxen gets pushed out. But if the FDA looks at some of the laboratory data and they go, well, you know, something here is just not quite right. Here we get a problem. So they're going to go, no, send it back to the sponsor so the sponsor can revisit, finish the problems, fix the problems, retest, and resubmit if that's what they want to do. Absolutely, 100% sounds legit, but it's not. And the reason is because at no point in time did the FDA go back to the sponsor or go back to the test facilities. These are the issues that we see in this flowchart. So pre-GLP, this is what was going on. So after these issues with IBT, the FDA says, you know, we probably need some regulations on good laboratory practices and how we're going to handle these types of situations and how we're going to prevent this from happening again. So this is post-GLP world now. So similar model, a slight change though. So I get a test facility, and that test facility can be, again, a local laboratory, hiring some people, maybe some of our graduates, doing some work, doing some tests. Absolutely. Everything's going according to plan. The sponsor is the people that will pay those test facilities. Sponsor's a bigger company, very often that's made this new product, that's done the clinical trials on the product, that thinks it's safe and thinks it's effective, but needs some third-party independent lab work to be done. So the sponsor pays the test facility does that job, and then the test facility sends the data back to the sponsor. The sponsor looks at that data, and the sponsor says, hey, this looks really good. I think that we're going to submit it to the FDA because I think this is going to be a product that's going to work for us. So the sponsor submits that laboratory data to the EPA just like they did before, but now the EPA, the FDA, or another regulatory agency, whoever they might be, they are now going to be required to go back and audit and investigate the sponsor and audit and investigate the test facility as well. They're going to have to brunt a little bit of the work. They're going to have to get out of their offices in Washington, D.C., or their local chapters. They're going to have to get out behind the desk. They're going to have to go out and investigate all of the lab areas and all of the manufacturing areas to make sure that these people are doing their job the proper way to prevent a problem like IBT had back in the day before this took place. So, external audits become a thing. That's what you're seeing down here. External audits, finally by the regulatory agencies, we're going to have to get required because those regulatory agencies cannot trust anybody. After they do their audits, after they inspect these locations, after they review the data that's been submitted, they then can still approve or decline these particular drugs that they're after, that they've been submitted, that they've been tasked to review. They can make better judgment calls based on that. All right? So, good laboratory practices, good laboratory regulations. A couple of things here with the regulations. Things that we can talk about, things that we've seen. No surprises here at all. Here's just a list of what auditors are going to look at in the laboratory setting. Again, very often if they look at it on the manufacturing side, they're going to look at it on the laboratory side as well. So first and foremost, they're going to review the organization of the laboratory and the personnel that work in there. They want to make sure they have proper training. They want to make sure that documentation is in place. All of the training records, the resumes, GLP training, SOP training, all of that is going to get reviewed by a regulatory agency because this is one of the ways to prevent an IBT from happening again. Next up are the facilities. I guess that's really properly number 2. So they're going to go and look at the laboratory spaces, again, sealing the floor, corner to corner, making sure that this is a clean lab, that it's organized and structured well, that people act like they know what they're doing in the laboratory spaces. Number 3 is the equipment. We talked about that already. All of the analytical equipment, including balances, hot plates, refrigerators, freezers, thermometers, you name it, they inspect everything. Number 4 is how the facility operates. So they're going to want to see the SOPs of the laboratory personnel because we're focused on GLP right now. Test control, references, substances, everything that you are using in the laboratory space to do the testing with, or the raw ingredients that manufacturing is going to be using. All of that's got to be reviewed. All of that's got to be documented. And 6, all of the reports and all of the records. So when you do the data, and when the data pops out, what do you do with it? How to review it? What goes on? That's records and reports. And an auditor is going to look at those things as well. All of these will help a future IBT from not happening. And I think that once you read that article and once you answer those questions concerning IBT, I think all of this will make perfect sense because this is what the FDA and the sponsors found out once they started to investigate the laboratory testing facilities and realize what a sham of things that were going on under that roof. But some people, believe it or not, picked that company to do the job because they knew how they were. They knew that they could get things passed with them. And as long as things got passed and they received that paperwork stating that it was passed, and then they could give that paperwork to a company like the FDA or organization like the FDA and get that approval stamp, that's all that this was about. So selectively picking a laboratory that's right for you, even knowing that what they were doing was shammy, it's not unheard of. And I guarantee you, it could still happen today. But it makes it much harder because of the structure that the FDA has in place now about cross-checking the sponsors and cross-checking the test facilities at the same time. All right, so that's where this little smidge of good laboratory practices will stop. In the upcoming videos, we're going to look at good documentation practices, which really go hand in hand with GLPs, and we'll pick up from there, and we'll talk more about how you should really spend your time in a laboratory setting.